Thank you so much for joining us today for this Taudia webinar. I'm going to go ahead and get started with introductions. My name is Jenna Gallegos, and I'll be the moderator today. Today, you are going to be hearing from Dr. Paul Hung and Dr. Ted Wilson. They'll explore biomarker detection with Taudia's NeuXplore technology. Paul is a serial entrepreneur with a track record of successful commercialization and exits. His companies CellAsic and Combinati were acquired by MilliporeSigma and Thermo Fisher, respectively. At Taudia, Paul draws on his extensive experience to turn cutting-edge science into market-ready products. Ted leads all fluid biomarker testing at Stanford's Alzheimer's Disease Research Center. His research accelerates the discovery of precision biomarkers and therapeutic pathways, advancing the fight against neurodegenerative disease. Without further ado, I will go ahead and pass it over to Paul to introduce the technology.
Hi, good morning, everyone on the West Coast, and then for the East Coast attendee audience, hope you enjoy the lunch and learn. And if you're from Asia, thank you for staying up late at night. Today, my name is Paul Hung. I'm the CEO of Taudia. Today, I'm going to talk about the redefining Alzheimer's biomarker detection with our platform technology, and then later on pass the ball to Ted to discuss his experience with the NeuXplore platform. Taudia's mission is to improve lives by making simple blood tests for brain health monitoring a part of the routine medical care. We would like to enable blood tests to better diagnose and better screening, better triaging patients at primary care, and then also for any of the research applications. Our vision is that we want to leverage the global PCR testing capacity where the world has established during the pandemic time to scale these blood tests particularly for brain health, neurodegenerative disease. And then we started by developing a platform technology we believe can be run on any PCR instrument. That's how we get the scalability. And then we definitely need a menu. And what we believe Taudia is only focused on neuro or brain health. You could find many different pathologies in the brain. It's still largely unknown, although we have making progress and breakthrough. You're probably very familiar with Eli Lilly's drug and Eisai's drug, amyloid clearing, and then the underlying principles of how the brain works, the pathway. All these becomes possible by new biomarkers being discovered and being detected and be able to follow and monitored. And this is our focus. I'll talk more about our Splash Precision Proteomics platform technology in the later slides and how what we believe is it has the characteristics to truly impact this field. And then, like I discussed earlier, we do not want to become a closed system where we have to work with our own instrumentation. We'd like to be able to have this chemistry work with a simple PCR platform that probably every lab has after 2020. And then also with various different sample types like blood draw or the dry plasma cards, or even the very popular right now, you have the micro capillary array that can hook up to your arm and then take about 300 microliter of blood remotely. And then we did not forget about software. Every customer, whether you're from academia, biopharma, CRO, or clinical, you will require software to be able to report out and then having the data behind it to make sense of the data and improve the prediction algorithm to understand what we're really detecting and the accuracy and precision aspect of it. Our current focus to improve brain health is on Alzheimer's disease, as we believe this is from the Alzheimer's Association Facts and Figures published in 2024. If we're not taking actions, there will be a significant economic and societal burden in the next few decades. And then on the optimistic front, we see that there are increasing number of late-stage clinical trials and also pharma companies started to invest in the neuroscientific or neuroscience area. I want to draw your attention to this paper published by Professor Oscar Hansen. It's from Lund University. It's a Nature Medicine review. It's a great paper on reviewing the biomarkers for neurodegenerative disease. There are new protein biomarkers being discovered, and then particularly on this graph, you see the biomarker trajectories in Alzheimer's disease that was in the paper. And then the AD biology and science slowly getting understood, definitely is benefiting the science advancement and also therapeutic development. So we believe this is the right place and the right time. However, this is published by Professor Schindler at the Washington University. It's also in Nature's Review Neurology last year. It's over 30% of the AD diagnosis in primary care are missed, which means it's false negative. So what happened is it was slows down the clinical trial recruitment and drug development, because how do we make sure like we're recruiting the right patients? And then also it delays referral to the specialists. Specialists are busy. They are a scarce resource. The neurologist has other type of important sort of like a disease or situation that I have to attend to. How do we enable earlier and then better triaging at primary care? And then, so what we're thinking about, what if actually routine blood tests, whether it's by hospital visits or dry plasma cards, or even just sending a kit to someone's home to collect the blood, that can improve the AD diagnosis accuracy and then the scalability and availability, accessibility. New technology is needed from our view. If we look at this publication again, to detect the disease early, we need better sensitivity. You could see the detection threshold. If it goes down, we'll be able to see the full picture when it started. So therefore, we will require a platform or technology that's very sensitive, mainly for early detection. AD is a chronic disease. It goes through these cycles, and then you will see things goes up and saturated, and therefore precision quantitation is important. Precision means, well, your CV has to be low. Quantitation means you have to provide an absolute value. In this case, the monitoring becomes meaningful. And lastly, well, if it cannot scale, it's not very useful, because if only a few labs in the world that can run this, instead of every lab in the world can run it and get consistent results, then it will not become a viable blood tests. So how do we design a solution that can meet all three requirements? And here I would like to introduce to you, this is Taudia's core platform technology. We call it SPLASH, a solid phase ligation assay with single wash. It started with you have to know your protein, know what you're looking for, and then there is an antibody that's able to bind to that protein. And then step two is you're going to form an immunocomplex by ligation assay. So ligation assay, this is first, well, I'll say patented by Olink Technologies, or actually the Swedish group from Lund University back in 2002. And then you have antibodies where you conjugate an oligo sequence to it. And then when the two antibodies both get in proximity of the protein of interest, these oligos can be ligated and become a PCR product. That's how you transform protein detection into a genomic domain using PCR. And then here you will see that we have a special orange dot in here. This is unique to Taudia. Is our technology actually other than, so you can see one of the antibodies, other than the oligos, we also conjugate a biotin molecule on top of that. And this is important because having that biotin molecule on one of the antibodies will allow us to do a solid phase action. So solid phase means using a magnetic beads coated with stravavidin. Now we can pull down these targets of interest and remove the background. And therefore, you have a PCR action, which is exponentially increasing your signal, and you have a solid phase wash action to release your noise, and you got maximize your signal noise ratio. Therefore, you have better sensitivity. And then and we can ligase and quantify using any qPCR instrument. Later on, I will show you this workflow is very simple with a single wash. It differentiates ourselves from like a typical ELISA. You have multiple wash steps, and the more steps you add into a workflow, typically science will tell you the precision, you're going to lose precision because every step you're going to add a little bit more variables into your process. And then with a single wash, we believe our precision is maintained. And later on, we'll show you that the data coming from internally and both from test experience. And lastly, a PCR software. PCR have a CT is a cycle threshold as of their main output. And then this can be converted into picogram per ml. This is very similar to, let's say, ELISA. You have colorimetric index, or you have an electrical signal from using MSD's platform or other platforms. You always have a reference point, and then you have to convert them into picogram per ml. So it's a similar concept here. It's not new. So going to the details. So the step one is a homogeneous incubation. So you have your sample, and then we throw in the sample buffer, and then you throw in your antibodies. In this case, you have the antibodies that only have the oligo, and you have the antibody that's dually conjugated. And after you incubate for, right now, our protocol calls for 2 hours of incubation. And this is just a biology need to happen where the antibody need to find the target. And then what we're going to see is now you in the same well, you're going to add your strep encoded beads. And this beads will start to, so biotin is going to bind to these strep beads. And also this serve almost like a physical barrier, if you see this, because you could see that if this purple antibody bind these blue antibody by chance and not specifically bind to each other, it creates a false positive signal. By putting a lot of beads around this, it decreases the chance of these two antibody not specifically meet each other. If they don't meet each other, the chances are they don't create that much background. So this is something that also a core technology Taudia develops, and then what we have proved that this actually effectively decrease the noise level. And then once you incubate your magnetic beads and capture your targets, and then now you could put the plate onto a magnetic rack and remove, just do a single wash. This is still zero transfer if between any consumables that stay in the same well. You're now taking out, you dump your supernatant, you wash, and then in this case, your background is removed. Also, in the same consumable, you started to add your PCR master mixes and ligase. In this case, you could find that even though there are some additional antibodies that was having about the dual conjugated antibody will bind to the beads, because it did not have a purple partner, it will not be able to form a PCR template. Therefore, it doesn't create a signal. Only the one that's captured onto the right protein target will be ligated and form the PCR product. And this whole consumable, still the same consumable all the way from the beginning to the end, you put it into, which is a traditional 96 well plate. And then you put it into a qPCR instrument and then use our software to analyze. So this is a screenshot of our software under development. Then you could design your experiment and then see the standard curves and report out picogram per ml on the software in a very straightforward way. So what's actually going to be in the kit? So this is what we're going to see. And when you get the kit is you have all your reagent needs to run up to 96 samples. You will have your protein standards, you will have your checkpoints, QC, and then sample buffer. And then for the antibody, you have a probe A and B. That's for the two different ligation assays components, the buffer, and then the magnetic beads to pull down to do the wash. And then Amplimix, it's the master mixes for the PCR reaction. Oh, this is a simple 3-step benchtop workflow to show you that this kit is going to go into the traditional PCR 96 well plate and then incubate the wash. The hands-on time is less than 10 minutes. It's mainly with a multi-channel pipette. You'll be able to confidently operate with the volume, which is 10 microliter, is what we believe. It's a very familiar lab workflow. Pipetting something very small volume is always challenging. You have to be very skilled to make sure you don't mess up. And then all these dilution and addition may also add additional complexity and impact precision. Using 10 microliter is what we found from customers. They're very comfortable with a P20 pipette. And then that match with almost all lab bench workflow. And you got a sample to answer in less than 3 hours. And then almost 100% sample utilization because it's always operating in the same consumable. This is the manual workflow. We have tested on multiple brands of very well-known brands of the qPCR instrument as well as digital PCR instruments. And then however, we also understand that for some of the labs, they prefer something that have a clear pathway toward like LDT or IVD. So we actually ODM this instrument. This is a sample to answer instrument. You can see here, it's about 110 centimeter wide. And then on the left-hand side is a liquid handler. And on the right-hand side, it's a PCR instrument. It's a dry system. You just load all the consumables and the samples and the reagents in there, and the system will automatically run everything and then report it out. The software, again, this is the PCR output using our same software. You'll be able to convert the CT to picogram per mil. So now let's take a look at the data. Does it work? So this is a phospholipid tau 217, very popular target. We have proved that we have a linearity down to femtogram per mil. You can see that even at about 10 femtogram per mil, we're still about 4 to 5 times above the background, while spike recovery is the industry standard 80 to 120%. And then, like we said earlier, early signal matters. So sensitive detection will reveal something other platforms miss. We also did a head-to-head comparison with one of the competitors' platform using the same antibody pair. We understand that antibody is very important, and then without using the same antibody pair, it cannot be really called a true apple-to-apple comparison. And using the same amyloid beta 42 pair, we found that we're about 5 times more sensitive than one of the competition. Consistency, with a similar idea, we acquired 20 samples from the biobank. It's nice 80 plus, 11 is 80 minus, or we want to do a concordance test with the leading platform in the market. And what we found is we call all the right patients from between our platform and competitors' platform. And then additional benefit is we found that all triplicates, our competitors has a CV up to 26%, and our CV is very tight within 7 to 10%. And precision matters, like we said earlier, every small change can tell a true story over time. And then we also have proved that we can do duplex assays at this moment. So p-tau 217, A-beta 42 simultaneously in a single reaction. And you can see that the performance of a single plex versus duplex are almost identical. And then we just throw this into healthy plasma samples. We can detect 100x above the background for both targets. So this has become our standard. What we believe is signal noise ratio is actually most important when it comes to ultra-sensitive detection, because you want to make sure you're well above the background, not just the absolute quantity of your LOD and all that. If your LOD is low, but your signal noise ratio is very poor, that's not really meaningful when it comes to a detection of a molecule of interest. So we have sort of like put ourselves in, how can we maximize this signal noise ratio? This is a growing menu. We have done the preliminary results on A-beta 40, GFAP, IO6. It's all well 100x above the background. And all these have a within place CV of less than 5%, even with just a single pipetter with a very skilled lab technician, you'll be able to get precision of less than 5%. And then we are actually working on NFL. We know it's a very important target, and as well as multiplexing up to 5 targets in a single reaction, which the team feel confident we can deliver, and that will match all PCR instrument capacity. Most of the PCR instrument have up to 5 optical channels, but we want to truly utilize those optical channels to be able to detect using single reaction, detect 5 different molecules is a 10 microliter sample. We are actively working with our collaborators to support assay migration and then custom assay development. There are actually 2 active projects. We have proven that this works really well. If you already have a biomarker of interest, you have an antibody pair, you have the protein calibrator and the gold sample, we could help you modify the antibody pair with the splash technology, assemble it, and then we'll run the calibrator check. If you let us know about the instrument of your choice, then we could run that and then put it subject to the gold sample. And then we review the sample, and then finally we can conduct the assay transfer to you. And then we have established this pipeline. We have tested this with two collaborators so far. It worked decently well. It proves that indeed our platform has a better sensitivity. And also for novel targets, we can very effectively turn around and efficiently turn around the experiment to prove that, hey, whether this antibody you're developing or this protein of interest really is the one that you should go target. So in summary, we believe this precision proteomics, brain health, neurodegenerative disease, these are all growing markets. That cross-section of proteomics and neuroscience is something what we believe is untapped potential. There's a lot unknown here. We hope to get more researchers, more scientists or pharmas, clinicians to help us grow this our understanding in this cross-section of proteomics and then neural disease. Particularly right now, we're focusing on AD patient screening, staging, and diagnosis and triaging. Our Splash platform technology is very sensitive, down to femtogram per ml with the full utilization of the 10 microliter sample input volume. Its precision is in the single-digit CV. That's great for longitudinal monitoring. And then it's scalable in the sense that it's fully compatible with your established qPCR or dPCR infrastructure. Or if you like to have a sample to answer system that's DX ready, we do offer that sample to answer boxes. And then we are launching our first suite of simplex and duplex assays at ADPD 2026, which is March 17th to 21st. Next year, March in Copenhagen. We have a booth with the number 39. What you are going to see is we have p 27, A-beta 42, A-beta 40, NFL and GFAP. Potentially have some more, and then as the targets. If you are interested to get a head start or learn more, our head of product, Jason Wang, his email is jason.wang@taudia.com, or you can email me directly, paul.hong@taudia.com for more information. Finally, I acknowledge the Taudia team and our advisors. Without their help, I wouldn't be able to present the data and the technology here with you today. So thank you. So please join us to accelerate the movement for simple blood test for AD triaging. Pass the ball to Ted, who's our next speaker. Thank you.
Thank you, everybody. I'm very happy to be here. Thank you, everybody for joining. My name is Ted Wilson. My lab is interested in studying the early events in Alzheimer's disease and the early processes that are associated with the disease. And one of the active forms of research in my lab is developing new biomarkers for the diagnosis of Alzheimer's disease and the early detection. And that focus has really been on blood biomarkers in the last few years. So I'm really happy to be here today to share some data on my experience evaluating Taudia's NeuXplore technology. And this is in participants along the Alzheimer's disease spectrum. And so this schematic gives us an understanding and sort of captures the complexity of Alzheimer's disease. And here are the key players. So we have neurons that show neurodegenerative processes. With neurodegenerative diseases, we also have involvement of immune cells, such as astrocytes, which are shown here. Capillary cells are involved in the blood-brain barrier, pericytes, for instance, and these cells begin to break down through the progression to Alzheimer's disease. And what this shows is that it's actually a complex process that happens, and overlaid with these cell types in the brain are the neuropathologies that are present. So we have the accumulation of alpha-synuclein that accumulates in Lewy body disease, Parkinson's disease. We have the accumulation of amyloid beta as plaques extracellularly. We have the appearance of tau fibrils, which build up as well. And then the third aspect that's overlaid on this image are the biomarkers that are present. And the biomarkers include things like neuroimaging biomarkers, like PET tracers, also biomarkers that are in the biofluids, like cerebral spinal fluid and in blood plasma. And what's really interesting about this process is as we learn more about the disease, neurodegenerative diseases, Alzheimer's and Parkinson's disease, we're identifying more and more targets that are present, and those have actually been developed to serve as staging molecules and biomarkers of neurodegenerative diseases. And so much so, you may be familiar with this slide, we've recently seen a revision to the biological and diagnosis of Alzheimer's disease. This is from Jack Jr. It's been recently revised. And in this case, what it's describing is how cerebral spinal fluid, plasma analytes, or neuroimaging might be used for the diagnosis of Alzheimer's disease. And so this revised criteria breaks biomarkers down into core 1 biomarkers that is sensitive to amyloid beta proteinopathy. That includes amyloid beta protein itself and amyloid beta PET. And then there are T1 biomarkers. These are also core 1 biomarkers. These are sensitive as well to amyloidosis, and that includes markers phospho-tau 217, 181, and 231. Core 2 biomarkers are sensitive to the tau proteinopathy. That includes tau PET and as well blood biomarkers, the new ones that have come online recently, MTBR tau 243, and other markers. And so what's interesting about this staging is other very well-known biomarkers that can be useful in Alzheimer's disease, although they're not specific to Alzheimer's disease processes, including neurofilament light and GFAP. These are used to shore up and provide confidence in the clinical diagnosis according to the the revised staging criteria. And this also includes biomarkers of non-AD co-pathology, so vascular brain injury so far, which can only really be assessed at the moment clearly using MRI and other metrics, and alpha-synucleinopathy, the S, which is sort of defined using the newly developed alpha-synuclein seed aggregation assays. And so these new revised criteria indicate that abnormality in core 1 biomarkers is sufficient for an Alzheimer's disease diagnosis. And that's really interesting when we think about it, but it leaves a lot of the biology sort of on the floor. And while the additional biomarkers are encouraged to give confidence in the diagnosis and to help with further staging, there really isn't a lot of guidance on how do we best integrate blood biomarkers. And this has been a question that my lab has been working on for some time. And what we see is that there's a possibility to integrate these blood biomarkers. And if we're able to do so appropriately, we'll be able to fully capture Alzheimer's disease co-pathologies. And so what we're finding, and this is supported by data from other groups, is that the order in which we think of the biological changes in the staging criteria may be out of sequence. And in fact, we see changes beginning with markers of amyloidosis, and then changes associated with the astrocytes, so GFAP, astrocytosis related to immune signaling, before we actually observe changes in p-taus, p-tau 217, so these being the core 1 biomarkers. And this is prior to overt changes in the vasculature as well. So we're continuing this line of research, and one of the things that we really need to do in order to address this question is to have high-performing, very sensitive assays that allow us to assess different biomarkers and incorporate new biomarkers as they come online to be able to measure these complex processes and these co-pathologies. And so Paul already went through the chemistry and the technology, but just to recap quickly, essentially we looked at the SPLASH technology. So it's solid phase ligation assay with a single wash. So it's a proteomic platform. The platform is sensitive. It's able to detect analytes down to femtograms per mil. It requires a low sample volume input of just 10 microliters. There's an easy single wash workflow, and it's capable of multiplexing up to 5 targets. And that's important when we consider the multiple targets that are needed for the measurement of these pathologies. In our lab, we've trialed the NeuXplore instrument. This is a fully automated instrument. As Paul mentioned, there's a liquid handler with magnetic rods that carries out the reaction here on the left-hand side, and that reaction product, after incubation and washing, is transferred to a qPCR instrument that is fully on board the instrument itself, and that allows a fully automated process and with around 4 hours turnaround time. And so we were able to access this technology and assess it in the lab. And this is some information just on the study participants. So I'd like to spend a little bit of time going through the makeup of the participants that we looked at. So we included plasma from participants who were healthy control or diagnosed with mild cognitive impairment or Alzheimer's disease. We had no significant difference in the mean age of the participants. Some differences in the number of female participants present. And at our research center, we actually have a fairly enriched percentage of ApoE carriers. So you could see here, we have 44% of our healthy control population is E4 positive. And that's just because E4 carriage is one of the topics of main interest at our research center here. The percent ApoE4 carriage is 38% in MCI and 80% in our Alzheimer's disease participants. We could see the MoCA score decreases with MCI and Alzheimer's disease. No significant difference in years of education. But we do see the percent CSF amyloid positive reflects the high amount of ApoE carriers in our population. So around 40% of our healthy control individuals, those individuals who are cognitively non-impaired, are amyloid positive. 75% of our MCI participants and all of our Alzheimer's disease participants that we looked at were CSF amyloid positive. CSF tau percentage is 11.1% in the healthy controls, increasing to 25% in the MCI and 87.5% in the Alzheimer's disease. And these measures were provided using the LumiPulse platform to measure CSF using cutoffs that we had previously established at our center. And so one of the data points that I'll share today is data looking at plasma p-tau217 using the Taudia NeuXplore technology. And so the first thing that we look at, so the first thing we consider in the lab when we're considering or evaluating new assays is the test-retest reliability. That is really key to us, that we see consistent measurement as we measure the sample within the run. So that's the same sample measured multiple times over the course of the same analysis run, but also over and between runs. So that the same sample tested between different batches shows similar performance. And so the performance on the first parts with the median within CV was 2.8%. So that's for plasma p-tau217. The assay has very low within run coefficients of variation, so that's very good. And the between run coefficient of variation is 3.2%. So that's also very low. These are very positive numbers in those cases. The reason this is important is because if the effect that we see due to amyloidosis, or the difference that we see with an Alzheimer's diagnosis between controls and AD participants is only in the order of 15% or 10%, a test that has a between run coefficient of variation of 15 or 20 or 25%, it would be unacceptable and wouldn't allow us to measure those differences. So here I'm showing the plasma p-tau217 results for our participants. On the left axis is the quantitative picogram per mil. That's on the y-axis. And this is shown median and interquartile range is marked for healthy controls, MCI, and Alzheimer's disease. And what you can appreciate is there is an increase in the healthy control moving to the MCI, to the Alzheimer's disease participants. And what we can also appreciate is that there's a distribution of participants. There's a distribution of p-tau217 values across participants in each of the clinical diagnoses. And this is what we would expect, keeping in mind that a high percentage of amyloid positive individuals in the healthy controls. And so what we do is we stratify participants according to their CSF amyloid status. So darker blue are the participants who are CSF amyloid beta positive. And what we can see now, the same participants stratified in this way, is there's good separation between the A beta positive individuals and the healthy controls. And this is really interesting because it shows that even in the healthy control population, so these are individuals who don't have symptoms, the plasma p-tau217 from Taudia shows good ability to discriminate amyloid status. High performance as well in the MCIs, although the cases are still a little bit low, and higher plasma p-tau217 values in the AD who are all amyloid positive. So we can look at these data stratified another way. So these are folks stratified by CSF amyloid and tau status. So the amyloid negative and tau negative show low p-tau values, and these levels increase with amyloid positive, tau negative, and the double positive group. And so what we can do is we can actually look at CSF amyloid negative and amyloid positive alone, and we can ask how well will the Taudia plasma p-tau 217 assay distinguish these groups, as we can do a ROC analysis. As you can see, the AUC is very high at 0.954. So it's showing independent of clinical diagnosis that Taudia's plasma p-tau 217 has high discriminative accuracy in detecting cerebral amyloidosis. And so this was really encouraging. We showed the assay has very low coefficients of variation. We see it increasing with clinical diagnoses, but it also has very high performance in distinguishing amyloid status. So the next bit of data that we looked at was how well it associates with clinical variables. And so in these data, we're looking at, or what we're showing here are the association between Taudia plasma p-tau 217 with MoCA. And again, we see high Pearson R, such that individuals with higher plasma p-tau 217 show lower performance on MoCA total score. And the next bit we looked at was how well does the assay associate with other established immunoassay platforms. And so what we did was we measured the same sample across the 3 different platforms. So here on the left, we have the fully automated LumiPulse plasma 217 assay compared to the NeuXplore plasma p-tau 217, and we see very high associations of Pearson R of 0.8. And we have as well the ALZpath, Samoa 217, and versus the NeuXplore, we also see very high correlations here as well. So encouraging results. So by way of summary then, Taudia's NeuXplore 217 shows very low within batch coefficients of variation. That was at 2.8%. And the batch to batch coefficient of variation was also very low at 3.2%. The assay showed excellent discriminative accuracy in distinguishing amyloid beta status. That's an AUC of 0.952. And we've seen strong correlation with clinical variables, including the MoCA, and strong correlation with p-tau 217 as measured using other technologies. So together, the assay shows high performance overall, and its flexibility and ability to incorporate new molecules, new biomarkers, its ability to be scalable on either the NeuXplore device or to use the manual workflow, and to be able to multiplex and combine data for multiple biomarkers in the same sample is very promising. So thank you very much. I'll be happy to take questions as we move on.
Awesome. Thank you, Ted, and thank you, Paul, for a great talk. Let's go ahead and start by talking a little bit more about the target patient and clinical environment for this test. At what stage of Alzheimer's disease do you expect will benefit most, MCI or maybe earlier? And then do you see it being used in a primary care setting or a specialty setting?
So it's been really exciting, the developments that have been happening in the biofluid diagnostic space over the last 5 years. It's been moving at breakneck speed. The advances are moving very, very quickly. What's also exciting is at the same time, the advances in the Alzheimer's disease therapeutics have also been moving at a very, very fast speed. And I think this is important to have both of those changes happening simultaneously. As the drugs that are being developed and approved for Alzheimer's disease improve, there will be an increased pressure and an increased critical need for us to have high-performing biomarkers that are reproducible, that are reliable, and that are early, that are very sensitive to the changes. As it stands now, there is a huge increase in the number of folks who are electing on their own to ask their primary care providers or their sort of neurology clinic care team to advise on where to access these blood biomarkers. And what we're seeing is that if that's not being made available to the patients or those who have concerns, these folks are going out and they're buying tests regardless. And the desire then is so strong that it's really something that ought to be put in place and move very quickly to clinical care and primary care, because some of the assays and some of the tests that are being accessed by individuals may have may not be quite as rigorous or might be a little bit more sort of low performing. And so I think the opportunity to have well-established tests and to have robust and reliable assays is really important, and it's going to change how we move forward through clinical care and how we enroll and how we get folks to their drug trials and how we get them into disease-modifying therapies. Also, how we identify folks earlier in the pathological state when the diseases or where the diagnostics or and the treatments will be most effective.
Paul, anything you'd like to add to that before we move to the next question?
Just a few things, and then thank you, Ted, and then for sharing again your findings, and then the thoughts on how we practice biomarkers, blood-based biomarkers in this field. This is just to share a few learnings because the team just came back from Alzheimer Association International Conference that's end of July, and then definitely there's a lot of movement in the blood-based biomarker space. And what we've seen is a single biomarker, for example, phosphate tau 217, can find applications in patient exclusion and screening, and that can do a very quick screening of a patient before they doing the enrollment in the clinical trials. Whereas probably everyone is already familiar with the FDA's approval of Lumipos, of the Fujirabio's p-tau217 over A-beta-42. It's a dual biomarker for symptomatic Alzheimer patients. And then there's a growing sort of like needs and demands or questions around like Alzheimer patient staging. Like we have for chronic disease like cancer, we have staging, stage 1, 2, 3, 4 at the primary care, or we can be able to make better assessment with which patient at what stage. Alzheimer is pretty much still in the beginning of figuring things out. Like one of the project, like Ted and I have been really discussing, is leveraging his learning about all these platforms and his map of all the multiple biomarkers. There seems to be some unique combinations that he's very interested in, like say, hey, maybe these combination can provide better staging for Alzheimer's disease patient. And we're very excited to support his effort, and together with potentially one of few sort of like a novel biomarkers that he discovered in his lab. So those are, I feel like there's still no definitive answer yet, but it's definitely a movement, like a test sites moving at very fast speed. And that's where we really need to be rigorous. It's not like a market opportunity. Everybody jump in and create a lot of noises. Science, we have to ground it on science. And to us, science is about repeatable results. It's about precision. It's about these highly sensitive platform offer repeated answers, accurate answers to the users. So that's why we like to sort of like throw out our name into the hat with a lot of people focusing on discovery, panel, just run the tests. But let's really focus on what Ted just said, the rigor, developing the assay in a way that it does test it against gold samples, give you as good results, and then the good old-fashioned science, which is spike and recovery, the curve, triplicate, all these things so that's where we see the field will advance by utilizing a technology like the Splash platform to better understand the science.
Interesting related discussion question. Do you anticipate that the invocation of AI is going to move the test from a screening test to a diagnostic test?
Fascinating question. So I think really what it's getting at, the question is how will AI be useful in improving or leveraging or advancing what the tests are capable of doing? And I think we're getting to that stage where we are going to be able to provide the AI or any model additional information, enough information. So when we have diagnosis or a staging framework that's based only on a handful of validated markers, there's a lot of nuance I think that is missed. And this is something that AI, especially when given sufficient data, is excellent at, is really digging deep within the structure of that data and finding interesting associations that may be missed. And so that's sort of the promise of precision medicine in this case, is that we will, with enough biomarkers that are identified, be able to winnow down a panel that isn't too broad with biomarkers, but is broader than perhaps what we currently are able to do, and really be able to hone in and stage participants and patients at this point, and really be able to capture appropriate diagnoses using those data. What else is interesting is there's an additional wrinkle here, is that there's an incredible opportunity not only for screening or diagnosing, but also for therapeutic monitoring. So as more and more folks elect to undergo anti-amyloid therapy, for instance, we still have a great opportunity to use these biofluid biomarker tools as metrics, as measures, to determine how well this therapy is working. How might the ARIA as a side effect of the amyloid drugs be mitigated? So these are the things that are coming forward, and I definitely think that there is a space for AI here to help contribute.
Yes, I completely agree with what Ted just said. It's the AI to us. AI is about if you can generate large amount of solid data, machine can sometimes figure something human cannot. And then it has proven in other field, and then we see this will happen in the life science field as well. However, again, how do we generate large quantities of solid data? And that's where also coming back to what Taudia would like to develop is something that when we think about large amount of data, we found that the large amount of testing ever done by human being is during COVID time. So PCR testing, probably we tested billions of people in that few years in order to advance the health and better understand the virus. What we believe is by using this platform technology can integrate into any PCR instrument in the world. Hopefully that can reduce the barrier of running the tests. And then with this chemistry, also improve the precision and the sensitivity of all these tests run on PCR instrument. And that amount of data, what we believe, can potentially move the screening test into a diagnosis test, because that's the large amount of solid data that we can acquire using this platform.
Taking the discussion of the impact of biomarkers in the clinic a little bit further, do you see them actually replacing PET scans, and if so, in what context?
That's a really great point. You know, they are complementary for sure. There are some aspects that PET scan are able to capture, especially when combined with MRI, that a biofluid biomarker won't be able to capture. Spatial distributions, hippocampal shrinkage, these types of measures. At the same time, the PETs, PET scans, while very, useful, they do have the drawback that they can be expensive. They're time consuming, and they also require access to a very well-resourced center. So the radiochemistry is complicated, and folks who maybe live in underserved or under-resourced settings don't really have access as well. So in this case, there is an opportunity for blood-based biomarkers to really eclipse what is done, at least in the early detection stages. And in events where, that data may not be clear or equivocal for the blood-based biomarkers, maybe in those cases, that's when you would refer for further follow-up using PET scans. But in these cases, the blood assays, as Paul mentioned in the beginning of his talk, they require very low level of resources to be able to collect those samples. Anybody who's undergone routine blood work is able to do so, and they are able to be measured in frankly any research lab that has a qPCR instrument, as Paul mentioned. So This is really important for the scalability of the assay and for the measurements. And I think we're sort of on the precipice of very, widespread assessment that's going to be happening. There's going to be a lot of interest from folks who are going to want to know their amyloid status or whether they are at risk or whether they are older or have a family history. It's a very intimate part of your identity, your memory and your mind. And so I think that really will drive a lot of interest, and the scalability is key because of that. And so the PET scans, not quite at the level to allow for that scalability. And this is where the biomarkers can really serve through the blood media.
This next question is for Paul. What assays are currently available, and what kind of customization or homebrew options might be available for the platform in the future?
As mentioned in the slide, so we are committed to launch the p-tau217, A-beta 42, A-beta 40, NFL, and GFAP, those 5 important neurological targets at ADPD 2026. And then so they are actually like Ted has been testing all the assays. And then so if you are very interested in getting an early start and then just try it out, very low barrier to entry. Like if you have like a qPCR instrument, you'll be able to run this assay. And then we're happy to reach out to us. And as for homebrew and customized solutions, as a very small company, you have to understand that we just don't have the resource to support that. Imagine we want to make sure the customers have a great customer experience. So therefore, what we like to do is if you have a unique target, please reach out to us. And like we already did with 2 other companies, therapeutic development companies, and then we could discuss how we can help you turn around your target of interests and then get ultrasensitive detection done fairly effectively. And by learning with these customers through collaboration and then of these novel targets, and then later on in the future, we may figure out a way to better support or have a high success rate of a homebrew kit at the customer's lab. But so far, the answer is we don't offer that, and then we prefer to work with the customer together so we can ensure we offer the great customer experience.
For those that might be interested in an assay for their target of choice, how long should they expect it might take to develop an assay for those targets?
So far, I'll say based on our current experience, we could complete the first phase, which is if someone already have the antibody, like I said, this depending on how ready the customers are. If you have an antibody already identified and they have a protein calibrator and then have a sample that we could test on, and then from our past experience, we can turn around the first batch of data in 2 to 4 weeks.
Awesome. For this next question, we'll start with your response, Ted. What's considered a good correlation between platforms, and how do you choose between all the different biomarker platforms out there?
That's a really great question. And this is something that we've been evaluating and working on since blood biomarkers have really taken off. So for us, the key feature really is the retest reliability. And, how well does the assay perform over time? So if you have an assay or a platform that shows low retest reliability, it really is a non-starter for us. The second aspect is we really need to have the sensitivity. We want to have an assay that is able to measure analytes in the preclinical stage of the disease, the pre-symptomatic stage of the disease. And so for that reason, it really needs to be able to distinguish between very, very small deviations in biomarker concentrations. The next thing that we look for is how scalable the assay is and the volume of blood sample that is needed. So by far the most precious and expensive resource that we have as a research lab is actually the plasma. The value of the plasma is the highest, the blood plasma, and even more so than the costs of the assays. So for us, it really is important to minimize that and to have something that is reliable so that we get the most out of those precious samples.
Paul, anything to add from your perspective?
What Ted said.
Awesome.
He's the customer. I don't even the platforms, right? We stand by our platform. What we believe is we are not the first to market, but we truly believe we're the best to market. And then hopefully what Ted said is with this ability to have ultra sensitivity, precision quantitation, and scalable with the global PCR infrastructure, we'll be able to advance the movement with the help from the customers and audience, and then to really advance the field, improving the therapeutic options and better triaging the patients. And then some team members, like including myself, have family members that sort of suffer dementia or some form of a neurodegenerative disease. So we really want to make this happen and then bring impact lives.
Awesome. Well, that is a great closing statement. We are out of time for questions today. If you guys did have more questions, feel free to reach out directly to the Taudia team via the website. Thank you, Paul and Ted, again for great presentations, and to you all for an engaging discussion. Thanks so much. Have a great day.